Hello, everyone, and welcome to Special Considerations for Managing Immuno-Oncology Studies, a New Paradigm, presented by MedPace. I'm Rebecca Williamson, the publisher of Fierce Biotech, and I'll be moderating this webinar. Our speakers from MedPace are Dr. Lyon Gleich, Vice President of Medical Affairs and Oncology, and Dr. Jennifer Cutter, Associate Director in Clinical Operations with a focus in oncology. You can read their full bios on the right side of your window. I've got just a few technical notes for you before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker if you want to participate in the interactive parts of the presentation. If you'd like to download the slide deck, you can click the Down Slides widget at the bottom of your screen. This webinar is being recorded, and it will be available on demand within 24 hours after the event. We will be following the presentations with a Q&A session, so please submit your questions during or after the presentation using that Q&A box at the right side of your window. All right, I think we're ready to begin. Jennifer, please go ahead.
Thank you very much, and thank you everyone who has joined this call. We are happy and delighted to be presenting this and discussing considerations for designing, managing, and executing immuno-oncology studies. I've been at MedPace for about 6 years, and I am focused solely in running and supporting oncology studies, and I have been directly responsible and overseeing most of the studies that have been done here at MedPace.
And this is Dr. Lyon Gleich. Prior to joining MedPace, I was an academic oncology investigator and was very involved in that time in immuno-oncology studies going back well into the 1990s. At MedPace, have helped lead and develop our oncology program for support of sponsors developing oncology drug products. And as you could well imagine, over the last few years, that's been taking more and more of a turn into immuno-oncology. We've been fortunate to have been at the forefront of that and to be helping our sponsors develop their trials, their programs, and to be able to see what works well and what doesn't, both within the U.S. and globally. I've been at MedPace since 2004 and have really enjoyed the efforts in the immuno-oncology space, leading in part to today's presentation. So one thing that we want to make clear is that this whole concept of immuno-oncology is not a new concept. Coley, going back to the 1890s, was proposing the potential for injection of bacteria and other products to try to stimulate an immune response. Decades ago, people tried BCG with some evidence of effects to stimulate the immune system. It's always kind of fun to talk about what came out after I finished medical school. It's fun to kind of realize that things like dendritic cells weren't recognized prior to my finishing medical school, and yet those have been given a Nobel Prize for their discovery. So the field's moving. It's an old field, but one that in recent time periods is now moving at a lightning pace and getting to drug approvals, and something that 20 years ago people thought was a dead field has now come alive. And we've seen lots of these type of things happen in oncology. We saw the era of cytotoxics. Then we've started moving into antibodies, then targeted therapies, and now immuno-oncology. So while I'm a big believer in immuno-oncology, I'm also a believer that it's not a be-all, end-all. We can see with the drugs that you get responses, but you don't get 100% responses in 100% of the patients. At the same time, there are a lot of opportunities for immunotherapy. This slide demonstrates the myriad of different approaches that can be taken with immuno-oncology, from using drugs that target T-cell-based therapies to drugs that stimulate the immune system to what's gotten a ton of the more recent attention, the checkpoint inhibitors. But I think it's important to realize that immuno-oncology is much more than just PD-1s. PD-1s are just one type of checkpoint inhibitor, and they're a type that does have some efficacy, and that's why the drugs are getting approved. But there are other drugs that target the immune checkpoints, like the CTLA-4s and other drugs in development with lots of potential. But there are also immune mechanisms that are being targeted and tried, some with quite promising data at this point. There are drugs with attenuated live bacteria that are being tried to stimulate the immune system, and some of those products even have breakthrough designation at this point, and those have nothing to do with the checkpoint approach. And then you can take all these different approaches, be it combining checkpoints or combining drugs that are different immuno-oncologic approaches together to see if that gets even a more robust response. So there are a lot of different clinical and scientific approaches one can take to this field. So vaccines have been around for a long time as one of the approaches. The vaccines might be utilizing specific peptides or proteins. There are dendritic cell vaccines being utilized. Some of these vaccine approaches require taking a sample of the patient's tumor or a sample of the patient's blood to extract antigens outside the body in a lab and then put that back into the patient's body either through stimulating the dendritic cells or stimulating other cells to try to then get an immune response. It's an approach that can work. It's sometimes a complicated approach, but again, if these approaches do get responses, they'll be of great value. You can do different approaches to activate the innate immune system. There are, again, a lot of different approaches for this to try to enhance antigen presentation. I already referred to the checkpoint inhibitors. The cytokine-type approaches are quite old. Using interleukins was done for a long time in melanoma, long before the PD-1s came out. It had some degree of response, but not the robust responses that we're seeing with the PD-1s and the CTLA-4s. And then there are adoptive T-cell strategies that are being tried where you take autologous tumor-reactive T-cells and inject those. It's a complex process, sometimes requiring external stimulation as well of the T-cells and then giving them back to the patients. And then you can also give therapies to block mechanisms of immune inhibition. So by no means when we talk about immuno-oncology are we talking about one approach, nor should the discussion of this topic be restricted to just PD-1s or checkpoints. All of these approaches are quite interesting and have long-term potential in the field of oncology. So when we're in a new field of development, which immuno-oncology in terms of getting drugs to the patients is still quite new, there's a lot of complex science tied in with complex patient selection. If we used the same old-fashioned inclusion-exclusion criteria that we used for our cytotoxic chemotherapy studies, we might end up with a lot of failed drugs. The old cytotoxic chemotherapy studies weren't necessarily trying to look for patients that still had a robust immune system. If you try putting somebody on an immuno-oncology study, but that patient might have had a burned-out immune system because they have bone marrow failure from their prior chemotherapies that destroyed their bone marrow, or maybe they're just very weak from their advanced disease, that might impact their response. Part of the problem is we just don't know. If you look closely at some of these labels, you'll see that some of the labels for the drugs that got approved do include companion diagnostics, but they don't include clear-cut testing for immune factors in the blood that we could theoretically utilize to select patients because none of them have proven to be perfect selection criteria as yet. Years ago, we used to try to do skin testing for tuberculosis as a sign of potential immune responsiveness. It's never been shown to be a great predictor. So it is one of the remaining problems for these drugs. Right now, you can take a very general number for some of the immune approaches that seem to work and argue that they probably work well in about 15% of patients. That means it would be great if from the start we could have a test that told us who's the 85% that don't respond or who's the 15% that do respond if you're someone that likes the glass full. Right now we don't have those tests, but I do strongly believe that if we use the same criteria as you do for a cytotoxic study, you're pulling in the wrong population. Right now, combination therapies are getting a lot of attention. The PD-1s have gotten approved and have a lot of excitement around them. CTLA-4 is approved. A lot of the drugs that are coming out in development right now that are using other approaches do have some mild to moderate activity, that there's thought that if you put this mild to moderate activity together with the PD-1, you might get even more robust activity. We're working with a number of different sponsors taking this approach. It's an interesting approach. It's a complicated approach because you might be using a new molecular entity that's one of the drugs being utilized. The second drug might also be a new molecular entity. The second drug might be one of the approved PD-1s, but maybe you're using it outside of one of the indications for which it's approved for, in which case it's also handled under an IND, and you have the issue of handling two separate drugs under separate INDs being referenced and needing a separate IND for the trial. MedPace has gotten experienced with these issues. They're complicated. The safety reporting can be complicated in terms of cross-referencing. It's a lot of different mechanistic issues when you're dealing with the two different sponsors as well to take into account. Sometimes it's convenient and one sponsor owns both drugs, but more often than not, that's not the case. Then you get the issue of what tumors do you do the studies in? You know, the initial studies were done in the tumors that are classically more immune responsive, like melanoma or renal cell carcinoma. But the PD-1s have been getting approvals in non-small cell lung cancer, which was clearly 10 years ago not viewed as one of the immune responsive cancers. So we do see, therefore, that there is a potential for the immuno-oncologic approaches in some of the cancers that weren't typically seen as immuno-oncology targets. Selecting the population there might be of importance. Even if we look at the lung cancer population, we know that the PD-1 responsiveness is higher in the patients that are squamous than in the patients that are non-squamous. We can subdivide that population. We know the responses are higher in the squamous population that have higher expression of PD-1 by companion diagnostic testing than the patients with lower expression. So you can keep subdividing these populations. This continued subdividing might be good because you might be able to get yourself into a population where you have a greater degree of response. At the same time, you might be backing yourself into too narrow a corner that you end up in something that's unrealistic to be able to recruit in a timely manner, especially with the amount of competition right now going on for drug development with immuno-oncologic products. With that, I'm going to let Jennifer make some comments as well as we get into site selection. I'm going to just start talking, though, as you're designing your study and getting to the point where you're ready to really think about going into the clinic with the product, or you've done your phase 1 in a broad solid tumor population, and now you're getting ready to go into a more specific population. It's extremely important to talk to PIs and thought leaders and clinicians in the field, and I would encourage you to talk to a mix of people. And what I mean by that is people that are at some of the larger institutions and are true experts in their area, be it a breast cancer expert if you're going into a triple-negative breast cancer study. But at the same time, I would think it's also frequently important to talk to some people in community practices to find out also how what you're thinking about doing from an academic perspective translates at the community practices, whether it's something that's a patient population they see and that they can participate in, or it's something that'll be more restricted to the academic centers. Or if you're thinking about going after a first-line population, you might start finding out from the academic centers that they don't really see that population as heavily because it's typically treated out by the community centers, and then having those community oncology input in the protocol design, since they'll be your main recruiters, will be very important. Additionally, an important issue with speaking to the investigators is the field of immuno-oncology is rapidly changing. Because if you were going to go do a study in lung cancer with an immuno-oncologic product right now, even if you talk to the top expert, they can't tell you for sure what's going to be 6 months from now. The PD-1s are still relatively newly approved. What their usage will be in different populations is changing day by day. There's the changes coming in from combination studies entering the market. So talking to the PIs, and we can help you with this, and we also have a good feel of this from the studies we're conducting and the frequent communication we have, we can help guide you for who to talk to as well as for what we're seeing happening. But it's very important to keep in mind that it's not just what's approved that you're competing with, but you're competing with these other combination studies that are out there, and you're still competing with the targeted therapies, the antibodies, and the cytotoxic chemotherapies. And if you're looking at an early-stage indication like a first-line, second-line indication, you're also competing frequently with standard of care. If you were in an end-stage population, then you're just competing with the study population, fortunately. So all of that has to be taken into account. Jen, can you also talk about how we do feasibility for the immuno-oncology studies?
Sure, absolutely. And I think one of the most important things when we're doing feasibility for a study is not just assessing if they have a freezer or the right equipment for it. We're really assessing also that they actually have the research infrastructure to support the study. This is a very competitive space. We have our coordinators that are very busy, our PIs that are very busy. So we also need to assess that they have the manpower to put towards your protocol. A part of how they get that manpower and all of that resources and shifted to your protocol is you have to make sure that they have interest in the trial design, but also in the product. So you're looking for a trial design that's going to be PI and site staff friendly as well as patient friendly. A couple of things that we can help with that is, making sure that that visit schedule is mapping onto standard of care for that patient, making sure there's not an undue burden for that patient to come into the site. And if there are additional visits that are required to put into place travel reimbursement or any other kind of mechanisms that's going to relieve that burden from that patient or from their family. So once we have all of that sorted out, we really do need to make sure that that site has the volume of the patients that are coming in there. If you are looking for a specific subtype of a specific type of a tumor, you have to overall see if they have that large volume of patient population that they'll then be able to pull out those specific patients that are going to meet your protocol. Again, the questions that we're asking at the time of feasibility, they're not always known on what is going to work well for this protocol. So you have to have very thorough, very specific questions, specifically getting at the patient population that you're looking for, and also to address any logistics for running that study. Importantly is also to look at any other competing studies that are out there, both immune studies and also traditional chemotherapeutic studies, because if there's another study out there that might have a trial design that patient is interested in, they're going to be offered all of those. So again, most of this is going to be done, and the CRO is MedPace. We can help with this. A lot of these discussions do happen at conferences between the sponsors and the PIs directly, and having these discussions directly with the key sites. And we can help with that with our medical expertise. We do rely on sponsors a lot and the relationships that they have had in their earlier studies and also through conferences. Lyon, would you like to add anything?
No, that's excellent. And we can't stress how important this issue of feasibility and planning is for the study. To recruit, one of the frequent questions we get from sponsors is, what are you going to do to recruit our study on time? And the two biggest things we can do to help with study recruitment are to help you make the best trial design possible to recruit the population with your regulatory goal, and secondly, then to pick the best sites to accomplish that. And things that we can do after that are much more limited, so that's why we spend so much time and effort on this issue.
Right, and again, I can't stress enough how key it is that you have that PI and that site staff buy into the trial design and make sure that they can actually execute the study at the site. Next one. Are we there? Oh, thank you. Again, when we're looking at site selection, we do want to look for sites that have experience in immuno-oncology, and those numbers of sites are increasing really by the day. If you are working with a site that doesn't have a lot of experience in running immuno-oncology studies, that's okay. We just really need to work with them and make sure that we are educating them so that they're well equipped to manage the study and also the patients. And one of the biggest things here, and I'll hand it over to Lyon, and one of the biggest things that's different between immuno-oncology studies and the cytotoxic studies that we're used to is really managing and handling of those adverse events.
So, with the immuno-oncology products, the adverse event profile changes, and we'll talk a bit more about that on later slides, but particularly having them prepared for the endocrine type effects and the immune type effects. Country selection becomes an important issue for the immuno-oncology products, especially with where we see things moving to right now. Many of the sponsors are trying to, at times, develop their immuno-oncology product, and for a variety of reasons, might want to recruit patients that have not yet been exposed to immuno-oncology products. So they might be doing a combination study of a PD-1 with a new molecular entity and specifically be requiring patients not had PD-1s before. If they're working in one of the indications in the U.S. for which the PD-1 has already been approved by the FDA, that can be quite tricky to fully recruit the study in the U.S. if patients can simply have already gotten the PD-1 as part of their standard of care treatment. And that would be a good example of when you'd want to be going potentially overseas, even if your number of patients required for your study is not that large. And your options overseas to be able to get populations where patients weren't already exposed to the approved products might be higher. Similarly, if you're trying to go to look at your drug before patients ever got cytotoxic chemotherapy and wanted to give your drug first line, that would also be a reason to really think about going ex-US. Obviously, the other common issue for going ex-US is going to just be driven by the size of the trial and the number of patients needed over the required timeline. There are a huge amount of similarities between Western Europe and the U.S. right now in terms of the handling of patients on immuno-oncology trials. And I would say that Central Eastern Europe offers some advantages in terms of some of these specific populations where there might be benefits to be able to pull those patients in. And Jennifer and I both already made some references to the importance of the site buy-in to the protocol. This really then filters down to the day-to-day recruitment of the study at the site level. The investigator being excited about your trial, the coordinator being excited is very important for driving the daily recruitment efforts for your study. We continue at MedPace to do everything we can to train the sites on the science behind these products and why the study is being done to help further push and support recruitment.
Yeah, one thing I'd like to add there, if there is a smaller study with fewer sites, again, that PI excitement and enthusiasm is going to be important for using their referral pathways to get the patients into their centers. For example, if you only had 20 centers open in the U.S., just make sure that they are promoting your protocol either at grand rounds or at tumor boards or providing updates at conferences is very important for getting those patients into the study and enrolling the study on time.
So here we wanted to talk a bit about important issues for trial design for immuno-oncology studies, and there's not a requirement to do the studies in a specific tumor type, but it's quite frequent from a regulatory goal. The sponsors are doing this, and we do, as we mentioned, see some of these studies done in extremely specific tumor types. So it's an important issue that you have clear what your regulatory goal is. Don't make the trial more specific than it unnecessarily needs to be because of the potential for competition when you're trying to recruit. Again, depending on the type of drug product you're working with, there may be a requirement for companion diagnostic. If that is the case, it's advisable to get work on that started as early as possible so that your studies are all done utilizing that companion diagnostic and the efficacy data can be readily compared from trial to trial to make decisions.
And I'll just add, from an operational standpoint, we have run into very complex trial designs, so we are tasked to make sure that we're getting the appropriate patients into the study. One of the ways that we can help mitigate that is putting together an enrollment form and having key criteria reviewed by the study staff and the sponsor prior to that patient being enrolled into the study, so we can ensure that we have the correct patient population in there. So that might include a pathology report. We could have, confirmation of the histological subtype, or even just confirmation that there is archived tissue available. And if that archived tissue is available, when will they be sending that in? So anything like that we can help with up front, just make sure that the appropriate patients are in the study. Again, if the trial design is not that complicated, we don't have to put this into place, but that is something that we utilize quite frequently.
And then we also run into a lot of special procedures typically in these studies, especially the earlier phase studies where the sponsor might be looking for a marker, a biomarker, a tissue marker to try to predict best evidence for efficacy, response, and safety. So we have run studies, particularly early phase 2s or phase 1Bs, that require tumor biopsies during the screening period, as well as follow-up tumor biopsies to see if the drug impacted the tumor tissue itself. We've had studies using PBMCs, in which case, as Jennifer alluded to during the feasibility, you need to ensure that the site can handle that. You need to ensure that they can ship promptly. If there's going to be a delay in shipping, you need to make sure they have appropriate freezer or refrigeration techniques. And depending on what's going to be run from the PBMCs, work with the central vendor to be able to have planning for what's the most amount of time it can be frozen prior to shipping. And also you need to take into account, if you're dealing with a global study, of what's happening with tissue that might be going from a European site to a U.S. vendor. Is there going to be a receipt vendor in Europe to handle that tissue and then make sure it's handled properly for transmission across the pond? Or is the testing going to need to be done in the U.S. and Europe at labs that use the same methodology?
So operationally, just making sure that there are robust procedures in place to know when those patients are coming in, when those samples are being drawn, when they were sent, and when they were received. So there's a lot of additional tracking that goes in because every single sample is important in these studies.
And then within specific criteria, there are tons of issues we could go into. The one that I pulled out to talk about today is just, in general, for these studies, you want to select patients that aren't on immunosuppressive agents and have the potential for immune activity. Some trials do include specific lab criteria, like maybe looking at the T helper cell levels. That's less common, but that does happen. But this exclusion of the immunosuppressive agents is extremely common, and steroids are often excluded, which can be problematic. The more advanced cancer patients are sometimes on steroids for pain management or management of other issues, and this exclusion can cut out a significant group of patients. So when you're thinking that, this type of tumor is common, and there are a lot of patients with this stage 3, stage 4 tumor that I'm going after, you have to think about these issues not just from the performance status requirement that you wrote into your protocol for what's getting left out, but also from these other criteria that are getting added in. How small are you making the potential population? As with any drug, you need to first demonstrate safety in phase 1. But then as you're going into the tumor types later, you typically need to do some degree of run-in to at least show that in that tumor type that holds. That can be just a very small group of patients. Sometimes 6 patients is acceptable, depending what's being done. If you're looking at doing a combination therapy afterwards, you would have to do some type of run-in or safety study to show that the combination of your drug with the other drug doesn't add any new safety concerns before you go into a more formalized efficacy study. And then I'm not going to get heavily into the biomarkers right now, but it's just an extremely important issue to keep in mind here because, of course, you're trying to see if there's a way that you can further show your efficacy and if you can pick out which patients aren't going to really be the responders. So with respect to safety, the adverse event profile of each immuno-oncology product is very unique. When these first started getting approved, the attention was being put upon the fact that these drugs would be hoped to be safer than cytotoxic drugs. I don't necessarily make the argument that they're necessarily safer. They just have a different profile. So you don't typically get the multi-week severe weakness and other issues that you can see with a cytotoxic. You might not be burning out the bone marrow like the cytotoxic might do, but you're introducing typically a very different set of reactions. There can be severe immune reactions that can be quite life-threatening and can be quite debilitating, especially for the short term. There can be long-term unintended immune reactions that occur, such as hypothyroidism, which at least hypothyroidism is typically relatively easy to treat, but that's if you made sure to diagnose it before it got severe. If you got severe endocrinopathy that affected the adrenal glands chronically, that can be a very big deal. So the important point here is that these drugs have a truly different safety profile and that they're not non-toxic. It's important as well in early phase studies to have clear plans with each site in case your product does produce in the early people that are treated unintended severe immune reactions. And if your drug has significant potential for efficacy, you might be going forward in development with a drug that you know produces severe immune reactions, and then need to have clear plans in the protocol, in the training of each site, for if these reactions occur, how do you treat them, how do they handle them, make sure the staff is well trained so that what might have been a dangerous grade 2 adverse event doesn't become a grade 4 adverse event with a very different outcome.
And part of that is also with patient education and part of the consenting process, that they're consented to know that even if something appears to be low grade, contact the study doctor, ask the questions, come in if needed, so you can mitigate that if it then becomes a more severe grade. Continuing on with the plans, we just highlighted here our safety plan. Looking at analysis for trends across the data, not only across the entire study but also within the sites. These are trends that are reviewed by the medical monitoring team as well as the safety team, but the operations team also gets involved if we're tracking for any dose-limiting toxicities or anything else that might be pulled out that wasn't necessarily expected for. Most of these studies that we work with do have an independent data safety monitoring committee or monitoring boards. So this is something that gets in place at the beginning of the study so they can help monitor this in a completely unbiased fashion. One of the things I would like to talk about is putting in place robust plans if you do need to have these longer safety follow-up visits. Early on in the study, patients are coming into the clinic more often, but then the visits will get spaced out. If you're looking at overall survival, there'll be visits every 3 months or so. But if there are visits in between for safety follow-up, maybe 100 days post that last dose, not the standard 28 days. Again, making sure that the sites have plans in place to follow that patient, have separate contacts or other members of their family so you can get that information from them. So again, upfront planning, robust plans will help the study go smoother. So, Lyon, I'll hand it back over to you to talk a little bit about the efficacy.
Yes. So, as always, if you talk to the Office of Oncology Drug Products and ask them what's their preferred response for an oncology study or an immuno-oncology study, they're going to tell you their preference is survival. That's always their preference. But we know that they're able to approve drugs off of other endpoints if they're well supported. And that can be PFS and it can be response, and especially for the earlier stage studies, you're going to typically be basing your earlier stage studies off of response in terms of making decisions. one of the interesting issues that we've seen with some of the immuno-oncology products is that despite a low response rate, they can show some significant impact on survival. So it's also important to keep that in mind, that just because you're not seeing a lot of CRs and PRs like you might expect with a cytotoxic agent, it doesn't mean that your drug's necessarily not working. Now, in terms of measuring response, we know that typically we're going to be using RECIST 1.1 in any oncology study, and we would still be using that in the immuno-oncology studies. But we also have the issue here of the potential for tumor flare, where the immune reaction in the tumor might cause the tumor to get larger initially. So that's one complicating issue that might make the traditional RECIST 1.1 not to be the endpoint that you want to use. And additionally, the response might not occur as quickly as with cytotoxic agents or even other targeted type therapies. So many studies have also used the immune-related response criteria. There are minor modifications of different immune-related response criteria that I'm not going to get heavily into. The biggest difference between the immune-related response criteria and the classic RECIST 1.1 is that in the immune-related response criteria, you're stipulating typically that new lesions are not automatically indicative of progression. So that is very important. And separate from that, a lot of the protocols don't call for taking the patients off study at the first progression, but allowing them to stay on study until either a second progression or clear evidence of lack of clinical benefit, so that the patients get the potential for more exposure to the drug in case they're having this initial tumor flare issue.
And Lyon, those are great points, and this is really something that we need to work with on the sites for the training and the continued reinforcement on keeping patients on study until they truly meet one of those withdrawal criteria. But I wanted to go back to the RECIST 1.1 measurements, and we're finding that having consistency in the site read of these scans is critically important. You have the same radiologist doing those reads and those assessments. We've had studies where we've gone back and done rereads and come up with different numbers. So, again, just making sure up front that you have a study-dedicated radiologist doing those RECIST reads.
So one of the other big issues with these studies that requires a lot of training on the efficacy, and when we're dealing with a typical oncology study with RECIST 1.1, even though RECIST 1.1 has been out for quite a while, we still end up doing a lot of training of site personnel at even the radiology and investigator level, reinforcing the RECIST 1.1 rules. When you're tossing in here the fact that you don't automatically take the patient off at first progression, it takes a lot of constant watching of the ongoing data and frequent communication by myself, our trial managers, our CRAs, to reinforce that to the site. And it's still quite frequent, even with large sites that we know are participating in a lot of immuno-oncology studies, where our CRA might be on the phone with them of what's happening with this subject. Oh, we took them off for disease progression, and then really they didn't have a prior one, and then, oh, we forgot, and getting that fixed, so to speak. So staying on top of those issues is extremely important because it's so habituated in many of the people at the sites that progression means come off study. And it's also important to let you know that I can't tell you how many times now I have seen patients on the immuno-oncology study where the partial response or complete response is not occurring until the patient's been on study for 6 or 8 months. So what we're reading in the literature about some of these late responses is very true, and there is real reason then for going to all these efforts of using these other criteria. In terms of writing the protocol for which criteria is the right one to put in your primary endpoint or as your key endpoint, it gets very confusing between the RECIST 1.1 and the IRRC, because I think they're both very important. You're going to want to be able to present what your responses were using IRRC, and you're going to want to still always be able to present what they were with classic RECIST 1.1. You might be using the second progression to allow them to stay on study and that's going to obviously affect your final survival analysis, but your RECIST 1.1 analysis would still be based off of traditional RECIST 1.1. So the way the analysis plan gets written to utilize this information is important. The way the protocol gets written to stipulate which might be your primary objective if you're dealing with a early phase 2 study where you're just trying to look for early efficacy and maybe your survival endpoint then is in your secondary endpoints, you do have to pick either RECIST or IRRC to be then your key primary endpoint if that's what you're after. And I do want to make clear there are other modifications that can be done of IRRC that we've seen sponsors use. What I put down has been the most common. And I do also want to mention that the tumor flare is also sometimes referred to as pseudoprogression, so that's terminology you see as well. With that, we'll talk a bit about regulatory challenges. Clearly, the regulators have been open to immuno-oncology. Products are getting approved. Products have breakthrough designations. So the FDA and EMA are both seeing the importance of this field and working with sponsors to develop the endpoints and develop the ability to get these drugs approved and to market. Further approvals will lead to even more clarity. We can look at the steps that some of the sponsors took to get their PD-1s approved, to get their CTLA-4s approved, to get some of the vaccines approved. And some of these drugs were successful at approval, but the PD-1s and CTLA-4s have been very successful at marketing. Some of the other earlier drugs that got approved were less successful at marketing. And that's also important to keep in mind when you're making your plans for approval is, are we staying in a wide enough population? Are we staying in something that's going to be heavily utilized, especially if the product might be expensive by the oncologic community? I've already made reference to combination therapies. Now, combinations can be combinations of immuno-oncology products, which I've spoken about. You might be trying to combine your immuno-oncology product with a cytotoxic, and that is sometimes done. It can be tricky to work out your timing for when it's best to administer them. You might want to work in animal models to figure some of those issues out first, and then during early phase human studies, figure out the timing as well, because there'd be concern about the potential immunosuppression blocking the potential response from the immuno-oncology product. Combining the immuno-oncology product with a targeted therapy or an antibody-targeted therapy is sometimes easier because you're not needing to worry about the potential immunosuppression, but you also have to keep in mind that antibodies in particular might be leading to an immune reaction that requires pre-medication with steroids. So you might then need to separate the timing of the two, and that's a different issue to deal with. So there's lots of complexities that come in with the combinations. I've spoken already about potential alternative endpoints to survival. Early discussions with the FDA and EMA can focus on these for what are acceptable in the tumor types that you're going after. The FDA will always require clear evidence of benefit. That's the language that's in the congressional laws that allow the FDA to have this oversight of oncology drugs. And this evidence of benefit is clearly survival is a clear benefit, but the endpoints that I talked about earlier, such as response, such as PFS, are potential other endpoints that can be discussed with the agency. If your drug is a drug that really has potential for clear evidence of benefit, then you do have the ability to also talk with the FDA about whether your drug qualifies for accelerated approval, Subpart H approval, therefore, or breakthrough designations if you've done a phase 2 that showed something that appears remarkable. And also, if you're going for something that's really new, where it's not a checkpoint approach, it's a different approach that the agency's not dealt with before, in that case, there might be value to the special protocol assessment so that the FDA has fully vetted and bought into your trial design as well as your endpoints but we do want to point out this can be a timely and cumbersome process, and it does not provide a guarantee that when your study's done that the FDA is required to grant approval. It just means that at the time when they granted the special protocol assessment, if you had the study done then and if the field remained unchanged, yes, they would grant you approval if it all finished that way. But things change a lot in between.
So just before we open it up to questions, I'd like to just summarize here. We've gone through a lot of the complex trial designs and the intricacies and logistics of running these studies. So it's very important at the beginning of the study to set realistic expectations around the startup timelines and when you're going to get first patient in, not only for the study but also by country. These studies are they can be very challenging depending on your drug product and the trial design. So there may be multiple levels of review as you're going through that startup process. And every site is going to be different. Every site has a little bit of different process there. And then the more complex or a new molecular entity that you have, the more complex your trial design is going to lead to multiple rounds of review of questions. So you're building your timelines, make sure that you take these into consideration. And then the last thing I really want to just mention is that when you're looking at your drug, make sure that the countries that you're going into have the ability to review your product so you're not wasting your time up front. So again, getting that advice up front, engaging the regulatory affairs teams, the FDA, the EMA, up front and early is extremely important. Lyon, would you like to add anything in closing before we open up for questions?
No, I think we can jump into questions because I know we've had a lot coming in. And Jeff is helping us with questions. So Jeff, let's, what'd you put up first? Good. So one of the questions was, in the preclinical assessment of combination therapy, including immunotherapy, should we transplant human cancer cells to mice with humanized immune systems? Do such models translate better than those using athymic nude mice? And that's going to depend a lot on the particular tumor type that you're dealing with and what existing models there are. It's not something with a clear-cut answer. You definitely want to do as much in the mice as you can ahead of time because it's obviously less expensive and ethically arguably better to be doing as much as you can in the mice ahead of time to predict your responsiveness than to be doing it in the clinic. But whether the athymics are better or the humanized are better, people go back and forth on all the time. Jeff, I know there were a few questions about tumor flare, so I'm going to just address that topic broadly. One of the questions I know is related to the timing of doing assessments given the fact that you're going to have tumor flare. Timing of doing assessments is always problematic. If you're looking at a cancer that might progress quickly, like pancreatic cancer or lung cancer, you don't want to make your time points 12 weeks out because you're going to have a significant percentage of your patients having significant tumor progression from their disease, not from tumor flare, in that time point. If you're working with a tumor that is less aggressive, then that's a bit different. If you're working in first line, it can be different than if you're working in third line. So on the whole, most tumor assessment timelines are typically 8 weeks apart. Some tumor types we see being done at 6. Some tumor types we see done being at 12. But with that as an understanding, then, if you are going to be taking the most common, which is the 8-week assessments, if you're dealing with the 8-week assessments, you might end up with the tumor looking larger at the 8-week time point. It might have been from tumor growth. It might have been from tumor flare or pseudoprogression. If you wrote the protocol as discussed that they don't come off for one progression, then they'd still be allowed to continue treatment. Your next scan, depending on how the protocol is written, because sometimes if there's evidence of progression, the protocols are written for the next scan to be done just 4 weeks later. So if it was written that way and you saw in the scan that was then done at 12 weeks that there was further progression, then the patient would come off study. If it was not written with the extra 4-week scan, that second scan wouldn't be done until 16 weeks. If you're seeing an additional progression at 16 weeks, there's very good reason to be taking the patient off. That's no longer likely to just be tumor flare. So that process tends to work around that issue if the sites are well educated. The other side point there is for some of these tumors, you do want to put some language in that if there's an extremely large increase in growth, that they come off without waiting for the second scan. Because tumor flare should only account for 10, 20, 25% increase in growth, not a massive increase in growth. One of the questions came on our thoughts on central radiology versus investigator site review. My answer on this is tinged with personal opinion. I am a huge believer in investigative site review. Even the FDA has recently published a study that they conducted along with a group of radiologists looking at drugs that have been approved in recent years and for whether, if they had not had central radiology review, the decision on any of those approvals would have been different, and the answer was no. So the FDA is arguably aware that they're wasting a lot of time and money with central review. At the same time, it's hard to tell you on your final registrational study not to play it safe if your main endpoint is not survival and not have central radiology review. But if your main endpoint is survival and you're going for approval off survival, there's probably no reason to have central radiology review. If your main endpoint for your final study is going to be PFS or response, then it's probably safest to have central radiology review.
Lyon, do you have any experience when a study has done a collect and hold on these reviews or on these scans? How many times they actually go back and review those scans?
So we've done a lot of studies where the sponsor was hesitant not to have central radiology and decided to sort of play it safe and do collect and hold. I've never had any of those go back and have them read. So it sort of reinforces the point I was making. One of the questions asked was, go back. Yes. Given the complexity of immuno-oncology trials, what trends in enrollment have we observed? So it's been influenced, in my opinion, more by a mix of the trial design as well as the population you're going after. If the population that's being studied is a population where clinicians think new therapy is truly needed, that it's a very underserved area, third line of cancer X that doesn't have an approved therapy, the early phase 2 data or late phase 1 data shows evidence of efficacy, even if the trial is complex, we've seen those studies recruit well. If the population that's being studied is earlier, where you're competing with standard of care, where you're excluding lots of things that patients might want to try before going on a study, then your recruitment starts getting much harder, and we can encourage the investigators to discuss the study with the patients much earlier in their course of treatment so that they have it in their thought process to consider the trial. But you have to then talk to many more patients at your site to get a patient to consent, and that puts a lot more burden on the sites, and those trials can be harder to recruit. And that's where we would then talk about having more sites, more countries, and the issues we alluded to earlier. Jen, anything to add on that?
I do not. You covered that nicely, Lyon.
A question was asked, in general, if a patient has one immune-related adverse event, are they more likely to have more immune-related adverse events and have greater severity events? And the answer is not necessarily. It's an extremely unpredictable area. We don't know why some patients tend to get more of an immune response and others don't. And by immune response here, I'm talking about untoward immune responses, not the immune responsiveness at the tumor. Nor have we seen anything up to this point that clearly correlates like it does with targeted therapies, where when we're giving the EGFR antibodies, we know getting the skin reaction is a sign it's working at the tumor. We don't have here the clear sign that if somebody's getting a severe reaction when the drug was given in their immune system? Is it clearly going to work better at the tumor? We do not know that. One of the questions is, in our opinion, can regulatory agencies accept an immunologic endpoint of the trial using immunocheck endpoint antibodies? So theoretically, they'd be able to. In reality, I would think it would be quite unlikely. Again, the FDA likes survival. Anything that they're using as the primary endpoint in lieu of survival needs to be something that's either already been used and accepted by the agency, or that you've gone to lots of effort to show them will be predictive of survival or a clear benefit. They're quite liberal in what you can use in secondary and exploratory endpoints, and the secondary endpoints can frequently make it into the label, but in terms of the true endpoint for approval, there is not a lot of wiggle room. One of the questions was, when studying combination products, how do you handle the adverse event attribution? So it's an important question. We deal with this frequently in combination oncology studies now. And we have some studies running where we're literally giving the patients 4 drugs at once. So when we're designing the adverse event form, we actually make separate columns for each drug for the investigator to independently assess relationship of each drug product. So if the study was using drug A and drug B, and let's say both are immuno-oncology products, and let's say both are new molecular entities that don't have a label and don't have a well-defined safety profile, if an unexpected event occurred, the investigator, and it was severe, and there wasn't another clear cause, the investigator might mark it as related to each of the drugs. If one of the drugs being studied is already approved and has a clear label, and this event that occurs is something that you almost never see with that drug, and the other drug being studied is new, they'll frequently judge it as related to the new drug and unrelated to the existing product. If the event is something that's very common in the existing product, they might judge it as related to that product, but then have to consider if they thought the new drug made it worse, and they might still judge it as related to the new drug. FDA understands these limitations and is accepting of that approach. You also get into the issue then of which of the drug products on the individual trial are investigational medicinal products, and in Europe, the issue of non-investigational medicinal products for background therapy as well, that can require additional reporting requirements that we can handle on your behalf and help you plan for.
So we're looking.
And that I hit already. We're good.
All right.
Any final comments you want to make, Jen?
I don't have any final comments, but thank you, everyone. We got some really great questions, and hopefully we have helped you to design and think about your next trial, and we would be happy to help you with that.
And it remains a very exciting area with lots of progress being made, and we appreciate your listening today and the questions that were sent in as well.
Great, wonderful. Well, thanks so much for those presentations. Really nice job there. And we want to thank all of the audience members for attending this Fierce Live webinar and submitting so many great questions as well. And I'd like to also thank our speakers for presenting, and MedPace for sponsoring today's webinar. The webinar has been recorded. You'll be able to access the recording within 24 hours on the same page that you used to register for the event. Thanks again for joining us, and we look forward to seeing you at future events.